Hello students, welcome back to YSR Academy. We move on to lecture 3 of reproduction in animals, and so far we have finished with the male reproductive system. We have done with the female reproductive system, and now we talk about the next part of the chapter, which is menstrual cycle. So let's begin with menstrual cycle. As we all know, we are a bit familiar to this because we have discussed in the previous lecture that the menstrual cycle, which is the characteristics of primates, one of the important features, including humans, involves cyclical changes in the ovary and the uterus, influenced by a group of hormones called as gonadotropins and ovarian hormones. These approximately 28-day cycle feature the release of an egg from an alternating ovaries midway through each cycle and are divided into 4 phases. So basically we have 4 phases of total of 28 days. Yeah, the menstrual phase, it occurs when the ovulated egg is not fertilized. Okay, fertilization nahi hua hai, causing the uterine lining to break down and discharge blood. Okay, it contains mucus through the vagina for about 5 days. This is the phase of bleeding. The breakdown is triggered by a set of hormones which are called as prostaglandins due to decreased progesterone, and then you have estrogen levels. During these 5 days, many primordial follicles develop into primary follicles and some develop into secondary follicles under the influence of FSH. So meanwhile, while the formed secondary ovum, which is a secondary oocyte, is released, you have newer set of follicles which will start to take up or develop, and that development of the newer follicles is influenced by a hormone which is FSH. Full form of FSH is follicle stimulating hormone. Okay, this is the graphical representation. So you have a period of 0 to 28 days, 14 being in the middle. Okay, so eventually the graph picks up at around FSH, peaks up around near 14, and then you have LH, which peaks up exactly at the 14th day, because LH is a hormone of ovulation. So without LH, ovulation is not possible, and without FSH, development of ovum, follicles, is not possible. Okay, ovarian levels of estrogen peaks up near about 14 days. Progesterone also peaks up later of 14 days, because then if at all this ovum which is released due to LH is fertilized, then you require that ovum to be implanted, and for implantation purpose you require progesterone. So you have these phases to consider. Okay, now we have the proliferative phase, which is the follicular or the post-menstrual phase, lasts from the end of the menstruation to ovulation, from 5 to 13th day of the menstrual cycle. The first day to say 5th day is the bleeding phase. Okay, it's a bleeding phase. Then we go on to say 4th to say 5th to 13th day is nothing but a proliferative phase. Okay, it's a proliferative phase. During this phase, 6 to 12 secondary follicles, develop, but only one becomes a Graafian follicle. While the atresia, जो नहीं develop हो पाए, those all cells would eventually undergo atresia. Developing follicles secrete hormones such as estrogen, influenced by gonadotropin, which is GnRH and FSH. The endometrium regenerates, forming endothelial cells, because you will have to now maintain the thickness. Okay, blood vessels form, reaching a thickness of 3 to 5 mm due to increased estrogen levels. So estrogen peaks up and maintains or increases the thickness. Then you have the third phase, guys, which is the ovulatory phase, which is the shortest phase. 14th day involves rupturing of mature Graafian follicle and release of the ovum into the cavity. So into the fallopian tube, there is a release. This egg is released through mature Graafian follicle. Process is triggered by rapid surge in LH. LH बढ़ गया. Okay, LH बढ़ा दीजिए, there will be release of the ovum out. The ovulation may be accompanied by mild or severe lower abdominal pain, because uterus and ovaries are contracting to push the ovum. Yeah, then you have something about the gametogenesis. Now students, when we talk about the gametogenesis, it's nothing but the process of two things. One, we talk about sperm, okay, and then we talk about ovum. Yeah. Now, so you have sperm and the ovum. So when we talk about development of the sperm, it is called as spermatogenesis. Okay. When we talk about ovum, it is called as oogenesis. So essentially, formation of sperm and ovum, let's combine it together and let's call it as gameto gametogenesis. Okay, gametogenesis. Okay, so you have a certain flow of spermatogenesis. Now, spermatogenesis is a process of production of the male gamete. Sperms are also called as spermatozoa from germinal epithelium. This we have already discussed because this is that epithelium or that tissue which is going to form germ cells. At puberty, the hypothalamus is going to secrete a hormone which is called as GnRH, stimulating increased FSH production or secretion, which initiates the spermatogenesis process. So FSH का बढ़ना और spermatogenesis को stimulate करना, ये connected or combined events है. Spermatogenesis occurs in the seminiferous tubules. We have already discussed where are they. They're present inside the testes, where germinal epithelium cell undergoes phases to generate the mature sperms. Now, this initially begins with the very first cell, which is your primordial follicle. This primordial follicle, guys, is diploid in nature. ये डिप्लॉयड है, इसको प्राइमोर्डियल फॉलिकल बोल दीजिए या फिर प्राइमोर्डियल सेल बोल दीजिए. इज गोइंग अंडर माइटोसिस टू फॉर्म 2 cells. सेकेंड माइटोसिस टू फॉर्म टोटल ऑफ 4 cells. मतलब एक ही सेल से नहीं बन रहे हैं वो, ऑब्वियसली माइटोसिस है, सो इट्स इक्वेशनल डिवीजन. एंड सेकेंडली वन सेल is dividing to form 2 cells. So now these formed cells are called as spermatogonia. These spermatogonia will be 2n, which is diploid. Now, one of the spermatogonia which is diploid is going to mature, grow, which is the growth phase, forming after growth primary spermatocyte. Primary spermatocyte. Primary spermatocyte is diploid, undergoes meiosis 1. Okay, now this is the meiotic division, which is a maturation phase. So undergoes meiosis 1 to form 2 cells called as secondary spermatocyte. The difference between the primary spermatocyte and the secondary spermatocyte is the former is diploid, the latter is haploid. So primary spermatocyte is diploid in nature, undergoes meiosis, which is a reductional division, to form secondary spermatocyte, which is haploid in nature. Undergoes meiosis 2, guys, to form spermatids. Spermatids will mature. Again, everything now will be haploid. So spermatids are maturing to form something called as spermatozoa, which will be N or haploid. And spermatozoa are the functional sperms. When you're converting or maturing spermatids to spermatozoa, process is called as spermiogenesis. And the entire process is called as spermatogenesis. There's a difference. Okay, so this particular primordial cell eventually divides, forms spermatogonia, as we have discussed, okay, which is nothing but a multiplication phase. So in which a primordial germ cell, which is 2n in the seminiferous tubules, undergoes repeated mitotic divisions to produce numerous diploid spermatogonia, which is 2n, containing 46 chromosomes. Yeah. Maturation of one of the cell to form primary spermatocyte, which is the growth phase. Again, it will be diploid. We have studied this now. Okay. Then finally, the maturation phase, wherein we have primary spermatocyte converting into secondary spermatocyte due to meiosis 1. Fantastic. And meiosis 1 follows meiosis 2 and formation of 4 haploid spermatids, which are non-motile and they are non-functional. Now they want to become transformed, they want to be functional. So how are you doing that? Convert them into functional spermatozoa. You are undergoing a process, and that process is called as spermiogenesis, okay, where they remain attached to each other and Sertoli cells. The process involves elongation, centriolar rearrangement, mitochondrial coiling, acrosome formation, and Golgi complex. So all of these things we will study in the structure of sperm. Yeah, which is the next topic, which is the structure of sperm. So essentially, when we talk about structure of sperm, it contains 4 different regions. Head, which is oval and contains a haploid nucleus. Now, because sperm is an haploid cell, so you have an oval structure, and this is the nucleus, which is haploid. Okay, above nucleus is a cap, and that cap is called as acrosome. Why is acrosome important? Acrosome contains lytic enzymes. Okay, they contain lytic enzymes. These are very important for entering into the membrane of the ovum during fertilization. Okay, next is the neck, which is a short region, contains 2 centrioles. Okay, the proximal and the distal. The middle piece, which is located or labeled there, contains an axial filament by 10 to 14 spiral turns of the mitochondria, which are called as the Neben kern. So this spiral mitochondria is also known as Neben kern. Okay, Neben kern, which is spiral mitochondria. Function, as usual, providing nutrition to the developing sperm. Or mobility. For mobility, you require energy to be given. Next, we have a tail, and that tail is long. Okay, it's tapering part with cytoplasm and an axial filament extending till the distal center. So it's a long tail. It's a flagellated one, so it's a propulsion movement. So it is going to move for sperm to propel ahead. Okay. Now we talk about the oogenesis, guys. Oogenesis is a process of formation of again ovum, okay, or haploid female gamete, okay, from diploid germinal epithelium, again same, involving both mitosis and meiosis. 3 stages में इसको divide किया हमने, multiplication phase, growth phase and maturation phase. 3 phases में रहेगा. ओके 3 फेजेस में. मल्टीप्लिकेशन फेज में अगेन सेम लॉजिक है. देखो कोई चेंजेस नहीं है. प्राइमोर्डियल जर्म सेल आपका ओके पीजीसी 2n रहेगा. माइटोसिस किया 2 सेल बना, वापस माइटोसिस किया 4 सेल बना. चारों के चारों सेल्स का प्लॉइडी विल बी 2n, which is 2n because they are mitotically dividing. Okay, mitotically dividing. Next we have the growth phase. Some oogonia will stop dividing, increase in size and form primary oocytes. During this phase, cellular organelles such as ER, Golgi and mitochondria increase in number. So there is a functional maturation to form primary oocyte. Primary oocyte is 2n, is going under meiosis 1. Okay, it is going under meiosis 1. meiosis 1 से 2 सेल्स बनेंगे. 2 सेल्स में एक सेल होगा जिसको आप सेकेंडरी oocyte बोलेंगे और दूसरा होता है पोलर बॉडी. So oogenesis begins before birth with primary oocyte entering meiosis 1 to form 2 haploid cells. 2 haploid cells बने. Okay, the secondary oocyte starts meiosis 2. meiosis 2 शुरू करता है but arrested at metaphase and is released from the ovary during ovulation. So ovulation में सेकेंडरी oocyte ही रिलीज होता है. ओवम नहीं. ओवम कब बनेगा गाइस? ओवम तब बनता है जब स्पर्म आने वाला है. ओके, स्पर्म उसी ओवम के व्हाट डू यू से इनकम्प्लीट मियोसिस को कंप्लीट करा देगा एंड फाइनली देन दे विल बी फॉर्मिंग अ कंप्लीट ओवम. सो मतलब ये नहीं है कि वो अभी जो ओवम से ओवरी से जो रिलीज हो रहा है वो ओवम है, वो एक सेकेंडरी oocyte है जो कि ये स्ट्रक्चर है. ओके, अगेन गोइंग meiosis 2 बनेंगे तो you will have polar bodies and there will be one functional ovum. Now there might be a question in your mind, what is a polar body? Rather understand it this way, polar bodies are nothing but a result of an unequal cytokinesis, which is cytoplasmic division. Okay, so out of the total of 4 cells produced, you have only 1 cell which is functional. Yeah, for provision of nutrients, for provision of all the essential things which are required for the development of the ovum is only given to the 1 cell. Okay, as opposed to spermatogenesis where all the 4 sperms mature. But while in case of ovum, we have only 1 cell which finally matures to form a functional ovum. Polar body is not a functional ovum. वो एक फंक्शन करने वाला सेल नहीं है, वो एक नॉन फंक्शनल स्ट्रक्चर है जिसका कोई भी रोल फर्टिलाइजेशन में नहीं है. सो ये चीज आपको जानना जरूरी है. ओके, नाउ यू हैव meiosis 2 which is completed upon fertilization, जो मैंने भी बताया, resulting in mature ovum. Okay, and a second polar body. If the fertilization does not occur, the secondary oocyte is shed during menstruation. Menstruation में ovum नहीं, secondary oocyte release होता है guys, याद रखना. Okay, this is the structure of a secondary oocyte. A human ovulated egg, which is a secondary oocyte, is about 0.1 mm, which is a non-motile cell, haploid with a large nucleus, which is called as the germinal vesicle. As you can see, the egg is surrounded by the vitelline membrane. Can you see this? The vitelline membrane, which is the cell membrane of the ovum. Okay, then you have the glycoprotein layer, which is called as zona pellucida, जो उसके बाहर है, and then you have finally the corona radiata. So आपको क्या मिलेगा? ये देखो corona radiata, pink color के cells. Then this layer, ये वाला जो layer, it is zona pellucida. The membrane of the ovum is called as a vitelline membrane. दोनों के बीच में vitelline membrane और zona pellucida के बीच में जगह होगा, उस जगह को हम लोग क्या बोलते हैं? Peri-vitelline space. Near to the nucleus, you have a very small reduced cellular structure that is called as the polar body. Yeah. So egg has polarity. The germinal vesicle in the first polar body जहाँ पे present है, उसको आपको बोलना है animal pole. उसके opposite को आपको बोलना है vegetal pole. Okay. Fair enough, students. I hope I'm not running too fast. If you feel so, please rewind. I'll try to make some changes if you feel. But what have we started so far? We have discussed menstrual cycle. We've finished with the menstrual cycle. We have talked about spermatogenesis. We have talked about development of sperm, structure of sperm. We talked about oogenesis. We talked about structure of ovum, which is a secondary oocyte, just now. Yeah. So moving on, which is the process of fertilization or syngamy. Again, we'll schedule it in the next lecture. I hope so. The video which I have covered so far, the topics that we have covered so far is well understood to you. Okay, and I assume that you're studying it. Okay, I will highly recommend, please make notes. Okay, please have a habit of making the notes, because that is going to help you later. We're covering the topic point by point, line by line, concept by concept. But if you don't remember it, if you don't know how to present it in the exam, trust me, it is of no use. Okay. I hope so. The lecture that we are, pushing are well understood. If you have some feedbacks, please get in touch with us, or maybe you can just put it down in the comment section. Any doubts? More than welcome. I will also, highly recommend you guys to subscribe to our YouTube channel, like the video, share the video with your friends who are have just started now, or maybe they are in between something. Just share the video with your friends. Maybe they could also get benefited, because we're covering each and every concept point by part and breaking down into multiple videos, especially only for you, because that will just enhance the way you learn, point by point, concept by concept. Remember concepts, रटना नहीं है, concept को याद रखना है, चीजें आपको automatic, समझ में आना start हो जाएंगी. With this, I'll see you guys in the next video. Till then, guys, take care, stay safe, and bye.